Hey, Hammy here, back with part 3 of section 6.1, where we will look at Mendel's second set of experiments, and then also sort of when we sort of discovered his work and some more laws and stuff that he discovered, more principles of genetics that he discovered or came up with through doing his genetic experiments with his pea plants. Okay, in the last video, we talked about Mendel's monohybrid cross, where he started with true breeding parents in the P generation, crossed them to make the F1 generation, and then let them self-pollinate to make the F2 generation, where he found a 3-to-1 ratio of dominant to recessive traits, or 1 to 1 to 2 to 1 of like a genotypic ratio of those traits. What he began doing, because he was following all 7 traits, is he wondered if traits were passed together. So if he followed more than one trait at a time, what would happen? Okay, we have since sort of termed this the dihybrid cross. Okay, so what if you look at the picture over here, what you're seeing up here in the parental generation up here, we're taking a pure dominant that is round seeds and yellow seeds. Okay, so you've got 2 traits there, seed shape and seed color. And over here is the pure line recessive. Wrinkle seed is recessive and green, wrinkled and green. Now, when he crossed them, all of the F1 generation, 100%, were what phenotype? Okay, can you figure that out by looking at the diagram over here? What gametes, if these are the gametes right here in the middle, okay, the sperm and the egg, what did the yellow, round yellow plant pass on? Well, it had big R, big R, big Y, big Y. So it had to pass on a trait, a dominant allele for each trait, big R and big Y for each one. Okay, pure recessive, of course, had to pass on a recessive allele for each trait. So all of the F1 generation are going to be what we call a dihybrid. Okay, so this is a dihybrid, di meaning 2. So it's hybrid for 2 traits. Okay. Now notice all of the F1 generation were round and yellow. Okay. Just like when we in the last video in the monohybrid cross, where he crossed purple and white flowers, all of the F1 generation was purple. Remember, there was no blending happening at all. Now, when he let them self-pollinate, you'll see that you get a whole different bunch of combinations. So down here are the gametes. So this side is the sperm. Over here are the eggs. Okay, so you could pass on a big R and a big Y, a little r and a big Y, big R, little y, little r, little y. And same thing with the other plant because it's the exact same genotype. And notice all the different combinations down here. And when he began counting seeds, he noticed a ratio. Okay, how many were round yellow? 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so 9/16. 9 of them were round and yellow. So it had both dominant traits. Okay, so if we do this as a ratio, how many of them were yellow but were wrinkled? Okay, 1, 2, 3. Okay, so 3 out of 16, 3/16 were wrinkled yellow. Okay, now how many were round but green? So dominant for shape, round. Okay, so 1, 2, 3/16. And I'll put this one down here so we're not all jumbling together up there. We're round but green. Round and green. Okay. Then if you notice, there was 1/16, 1 out of the 16 of the possibilities here that was recessive for both traits. So it would be wrinkled and green. Okay, so we don't do the genotypic ratio in this because it would be kind of messy. But as far as the phenotypic ratio, we would say phenotypic ratio. Okay, phenotype, and we'll get to a little bit later in this video how they look. We say in a dihybrid cross, Mendel noticed a 9 to 3 to 3 to 1 ratio. 9/16, about 9/16 were round and yellow, dominant for both traits. 3/16 were dominant for one, recessive for the other trait. And then 3/16 flip-flopped, recessive for the first one, dominant for the second trait. And about 1/16 was recessive for both traits. And he found this when he started following more sets of one, and he collected a whole bunch of data in his notebook. Okay, because he got this 9 to 3 to 3 to 1, okay, what that told him is that, so of this dominant trait, if you take two of these numbers together, what's 9 plus 3? 12. And these two numbers, 4. Okay, 12 to 4 simplifies to 3 to 1. Okay, or if you take the other trait, dominant and recessive is 9 and this 3, and then this 3 and this 1 is 12 to 4 or 3 to 1. So he said, you know what? The traits are passed independently of each other. He called this the law of independent assortment. Now, he did not know about meiosis yet and gametes and that kind of stuff. So, but he figured the factors, or what we now call genes, okay, the genes for the traits, or factors he called them, were inherited independently of each other. So we now know in meiosis, okay, when the homologous chromosomes line up, we talked a little bit about this in that chapter, how you can take your maternal and paternal chromosomes and they can line up on either side. Okay, so if you look at the homologous chromosomes, so here's trait F on these two, and here's trait W down here on these, whatever that would stand for. Now notice, here they have both maternal on the same side and paternal on the other, and it's showing two different options. They could also assort differently. So here's maternal on the left, paternal on the right. Down here they switched them. They put paternal on the left, maternal on the right. So that gives a different combination. And then when they go through meiosis 2 and they make the possible gametes or sperm and egg down here, these all have big F, big W's, a big F and a big W, or a little f and a little w. Over here, big F, big W, or little f, little w. Okay, if you remember, it looks a lot like what went down the side of our little possible outcomes in the last, the sperm and the egg in that last slide. And you can rewind and look at that. Okay, that Mendel said is because the genes are passed independently of each other, and they can be in that random assortment, and you get an equal chance of inheriting any of those genes from the parent. Now, Mendel put a lot of his work together and published it in 1866 in a book called Experiments in Plant Hybridization. Okay, down here is kind of a photocopy of that book. Okay, not in English, obviously, probably in Dutch or German. You know, remember he grew up in Austria and was in a monk in Czechoslovakia. Okay, so, but why didn't, at the time, Mendel's work wasn't like, oh, this is awesome, and everyone talked about it. It was largely ignored. And, why was that? Well, part of it might be that Mendel was at a remote monastery, so the lines of communication, his ability to travel and explain his work at different conventions was probably limited. And so it was, largely ignored for about 50 years. When in 1900, 3 botanists or scientists independently, in crossing different types of plants, came up with similar ideas. And then they were looking through, different literature, and they stumbled upon Mendel's book here. And they're like, oh my goodness, this guy had it right 50 years ago. Okay, and so it wasn't until the early 1900s that Mendel really got his due. The other interesting thing, side note, happening at this time is in the mid 1800s, there was another very famous person in science that we're going to get into a little bit later here this year, okay, by the name of Charles Darwin. Okay, that's right. Charles Darwin was working on his theory for evolution in the mid to late 1800s when he published his work. Okay, again, Darwin did not know about genes or some of these patterns of inheritance. And some people think if these two, Darwin and Mendel, would have connected back in their time, they would have advanced this idea of genetics and that kind of stuff greatly or tremendously, because their work really would have complemented each other, and it would have helped Darwin explain some of his evolution through natural selection and some of the mechanisms that we now know behind that. So kind of an interesting side note about two famous scientists in the areas we study in Biology 1, that were kind of doing their work, and it would have been kind of cool if they would have met or talked to each other on their work or their findings. If we fast forward, 50 years, 100 years, some current terminology is what I sort of want to finish with here today. We now know that what Mendel called factors of inheritance, okay, I mentioned this earlier, we now call genes. And he said that they are inherited independently of each other. We now know that's because all of Mendel's characteristics were on different chromosomes. Okay, and then later we began to understand that those chromosomes, those units of inheritance, are actually made up of this genetic material we call DNA. Once we get to the mid 50s, 1950s, when we discover that. Okay, the position of these factors or genes on a chromosome is called its locus. Okay, its locus. So sexually reproducing organisms have two copies. And remember, we call those two copies are on homologous chromosomes. Okay, one would have come from that organism, that individual's mom, and one would have come from its dad. Okay, one from each parent. So here is the locus for flower color. So at this spot on both chromosomes, on both homologous chromosomes, there is the gene for flower color. Now, that gene can have different alleles. Okay, and this is where we talked about dominant and recessive and so forth. So from mom is passing on an allele for white flowers, where dad in this case is passing on a gene for purple flowers. Okay, and then it depends, you take those two genes, and remember, purple was dominant over white. Okay, so this would be a purple. This would be a hybrid right here because they have an allele for a dominant and recessive trait. Okay, and so when they have offspring, they have an equal chance of passing on the dominant gene over here, or a 50% chance of passing on the recessive allele over here. And we now understand that through the idea of meiosis, gamete formation, and this idea of genes that are located in the DNA or chromosomes. Finally, I just want to mention some terminology just to make sure that we are clear on this. I've used these terms in the last couple of videos. We want to get definitions down in your notes so that you can refer to them and see some examples so that we make sure we understand these terms. Okay, the genotype of an organism is, I like to think of genotype, their actual genetic makeup. So the actual alleles that an individual inherits, what forms of the gene do they get? Okay, and we often use terms like homozygote, homozygous, or heterozygous with that. For example, if they have two dominant alleles for purple flowers, okay, here they're using capital B. Okay, we say that is homo, which means the same, homozygous, dominant. Okay, so remember the dominant allele is the one that's going to be expressed. Okay, it can cover up or mask a recessive allele. If you have a dominant and a recessive allele, hey, get out of the way down here, or a recessive allele, okay, then we say you are heterozygous or a heterozygote, or we say you are heterozygous. Okay, sometimes we call this a hybrid, right? Okay, now notice the dominant gene will mask or cover up the recessive gene, so it will still have purple flowers or show the dominant phenotype. I remember phenotype is the actual physical appearance. Physical appearance. So you could have two different genotypes, homozygous dominant or heterozygous, and you will still have the same phenotype. Different genotypes, same phenotype because of the whole dominant recessive thing. Okay. And then down here, the third possibility is that you have two of the same recessive. This is homozygous recessive. Okay. And the only way you can get a recessive trait is if you are homozygous recessive or you have two recessive alleles. Okay, and that will give you the recessive trait. Okay, so just some terminology, and we'll practice these. We'll work through practice problems with these. But this kind of gives you an idea of kind of how Mendel sort of stumbled upon more than one cross at once, and some of the terminology that we now use to describe his crosses.